My name is Sæmundur Ro. I'm a medical doctor at Landsspítali, the National University Hospital of Iceland, and I'm a PhD candidate at the iStopMM team in Iceland and been a part of it since 2016 when we started out. I'm here at ASH this year presenting my findings on monoclonal gammopathy of undetermined significance and the significance of that in the era of COVID-19. Our key findings are that MGUS, or monoclonal gammopathy of undetermined significance, does not confer an increased susceptibility or increased risk of severe COVID-19. Those findings are really reassuring because we have many reasons to believe that MGUS would increase our risks or our patients' risks during the time of COVID-19. When we started out in 2016, we didn't dream of this study, but when the pandemic hit, it very quickly became clear that patients with myeloma had an increased risk in the COVID-19 pandemic, and we thought that we should leverage our data that we were already collecting to answer this pressing question in MGUS. So MGUS is the precursor to myeloma and has been associated with immune dysfunction, has been associated with thrombosis, renal disease, and infections in general. And you would believe or would think that that would increase the risk in COVID-19 as well. And many have actually thought that. But we could actually answer this question in the iStopMM study and reassure our patients that actually MGUS itself is different from myeloma. It's a less severe disease. It doesn't confirm the same risk in COVID-19. And I think those findings are really important for patients and highlight the differences between the precursor and the full-blown disease. We started out in 2016 by inviting everyone in Iceland age 40 and more to participate, and we got a great participation rate of more than 80,000 people. And then we collected samples passively. So people came into their healthcare center or the ER for something else, and we got an extra blood test where we could extract serum and send to the binding site, where the people of binding site performed a serum protein electrophoresis, free light chain assays, and also assays for immunoglobulins. It's through these tests that we can really identify a population and a unique population of individuals that don't know that they have MGUS, are truly asymptomatic. So they are a screened population. So taken together, we did population-based screening at the binding site, looking for monoclonal proteins or immunoglobulins and abnormal free light chains using the free light assay. I think that mass spectrometry is really the next step in providing accurate measurements and accurate characterization of monoclonal proteins. I think that it's clear that the serum protein electrophoresis and the free light chain assay really just shows us the tip of the iceberg, and the mass spectrometry can really move the field forward in accurate detection of these monoclonal proteins. I do, however, think that it's an important caveat to that is that we don't know the clinical significance of the bottom of the iceberg. So I think that will be one of the important things moving forward in iStopMM, is to really characterize what does this bottom of the iceberg mean. And I think there is probably some clinical significance, but we need to elucidate that further before we can move it into clinical practice in this space. And I think that's what we're doing.